wages. Thank you for attending this webinar entitled Translational Cardio-Metabolic Medicine from Bench to Bed. This webinar is hosted by Pokja Cardio-Metabolic Department of Internal Medicine, University Gadjah Mada, Sardjito Hospital, Yogyakarta. Today we will have some presentation by distinguished international speakers in the field. But firstly, I would like to invite Dr. Prenali Dwisthi Sattwika, internist, as the moderator for the first session. For Dr. Prenali, the time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Dear colleagues, welcome to today's webinar from Cardiometabolic Working Group, Department of Internal Medicine, Faculty of Medicine, Public Health and Nursing, Universitas Gadjah Mada, in collaboration with Kobe University and Gunma University. The title of our webinar today is Translational Cardio-Metabolic Medicine from Bench to Bed. For the first speaker, there will be Prof. Noriaki Emoto, MD, PhD, and I have the pleasure of being joined by him. And I would like to introduce him first. Prof. Noriaki Emoto, MD, PhD, is a professor at the Division of Cardiovascular Medicine at Kobe University Graduate School of Medicine and at the Laboratory of Clinical Pharmaceutical Science at Kobe Pharmaceutical University. After his residency as a cardiologist in Kobe University Hospital, Japan, he conducted his graduate studies and postdoctoral fellowship under the supervision of Dr. Masashi Yanagisawa in Dallas, Texas. He won the Louis N. Cass Basic Science Research Prizes of the American Heart Association in 1994 for the molecular identification and characterization of endothelin converting enzyme ECE1 and ECE2. He moved back to Kobe, where he leads his own laboratory as a physician scientist. He is in charge of the pulmonary hypertension group in Kobe University Hospital. His major research interest is the translational research of endothelin. He is a member of Endothelin International Advisory Board and also a member of a board of directors in Japanese Pulmonary Circulation and Pulmonary Hypertension Society. For this webinar, Prof. Noriaki Emoto will be delivering a lecture related to hypertension and circadian rhythm. And we strongly encourage you to actively participate in this webinar by sending your question or comment at any time during the webinar through the chat. And we are going to discuss the questions later once the presentations are done. I will hand over to Professor Noriaki Emoto.
Thank you. Thank you very much, Dr. Prenali, for your kind introduction. And
Professor Noriaki Emoto, the time is yours.
Thank you. And good afternoon, ladies and gentlemen. First of all, I would like to thank this opportunity to give a talk. And at the same time, I was a little bit regret that if I can go there, it will be more fruitful discussion. But anyway, I would like to give some glimpse of our study. So let me share my slides. So can you see these slides? Okay.
Yes, please.
Thank you.
Yes, already, Professor.
Okay, thank you. So before starting my talk, oops, yeah. before starting my talk, I would like to introduce my Indonesian fellows. I mean, since 1997, we have been working on many Indonesian students, and now they are not students anymore. They are graduated to get a PhD, and they are actively working and in Jogja and the Jakarta, Malam, several places in Indonesia. And these people are red color are from Gajah Mada University of Gajah Mada. And our moderator of the next speaker, Dr. Professor Kitamura, Dhite, was working in our laboratory. So let me start my talk. This is the onset of the myocardial infarction in two different studies, Mill study and ISAM study. And as you can see here, the onset of the acute myocardial infarction has some diurnal variations in the later morning time has higher incidence of acute myocardial infarction. So this is not only for acute myocardial infarction. For example, the bronchial asthma also have clear diurnal variation, or vasospastic angina also have a tendency to occur in the early morning. So the onset of the disease have some diurnal variations. And of course, the blood pressure has also a clear circadian rhythm. And those abnormal circadian rhythm of the blood pressure may cause some disease. For example, this study, this is rather older study, but published in Circulation, that the sudden increase of the blood pressure in the morning, this is called morning surge, have a highest correlation with the stroke. So the abnormal circadian rhythm or abnormal diurnal rhythm may cause some disease. So based on these, we have been interested in the circadian rhythm and molecular clock because in 1997, the genes consists of the molecular clock has been identified. Later on, those people who discovered the clock gene got the Nobel Prize. So as you can see this picture, all mammalians have their own clock, and those clocks are in the brain. So even for the rat or mice, they don't have clock, but they have a clear circadian rhythm, and which shows like almost 24 hours. In the case of human, the internal circadian rhythm is almost 25 hours. So what is the clock? The clock is located in the small nucleus in the brain. It's called suprachiasmatic nucleus. And this is the central clock. It's like oscillators. So this is the real clock. And those clocks are controlled by some input or affected by input. And the biggest input is light. And because of these oscillators, there are many outputs, including physiological activities or behaviors. So those oscillators and input and output are the main important component of the biological clock. So again, almost 20 years ago, we have been working about the molecular clock in the cardiovascular system. And the first report from our laboratory in circulation is that we identified clock gene oscillation in aorta. And those, even in the cultured smooth muscle cell, have the clock genes, and those clock genes are, the oscillations are induced by angiotensin II. So previously, people believed that the clock is located in the brain, and this is the only clock in the body. But actually, including this study, many studies have shown that the central clock is here in suprachiasmatic nucleus, but all organs or even all cells have a set of clock genes. So this is called peripheral clock. This peripheral clock is controlled by central clock, and it causes their behavior like sleep, awake, cycle, or physiological functions like blood pressure control, heart rate variation, or body temperatures. So what kind of molecular mechanism causes the clock oscillation? This is a very complicated system. So there are several transcription factors which cause the oscillations. And those per gene or BMAL1 or clock gene or cry genes are very work together to make almost 24 hours oscillation or cycle to give an output function. So those abnormality of the clock gene is really involved in the disease. So this is one of the example to show the importance of the clock genes in human disease. This is called familial advanced sleep phase syndrome. You may not hear this disease before, but this is rare, but this is a condition that is characterized by a recurrent pattern of early evening, like 7 o'clock in the evening or 8 o'clock in the evening, sleepiness, and early morning awakening. So there is such a kind of family members. So this black circle or black square, these people tends to sleep early and wake up in the midnight. And one of those people has been wondering if she is okay or not. And one day, her granddaughter told her that granddaughter has a concern that she wake up in the early morning and she feels very sorry. Then that grandmother thought that, oh, this might be disease. And she consulted many psychiatrists. And finally, one of the psychiatrists found that one of the clock gene mutated in these family members. So this shows that the clock gene has not only clock gene is important for not only physiologically, but also may cause disease. So what is circadian rhythm for? So one of the answers might be adaptation of internal environment and outside environment. And if those maladaptation occur, then it may cause diseases. This was our hypothesis. So we set up two questions. One, will abnormal circadian rhythm worsen the prognosis of cardiovascular disease? And the second one is, will collection of abnormal circadian rhythm improve the prognosis of cardiovascular diseases? So the first one, this is one of the publication in Nature by Darwin et al. And this publication, this is rather cardiovascular disease. So when the normal wild type mice give a stimulation of the tachycardia, there is no change in the EKG. So the arrhythmia would not be induced. But KLF15 neural mouse, this is one of the transcription factor neural mice, shows that with those stimuli, it caused a fatal arrhythmia, ventricular tachycardia. And wild type, there's no such a case, but this mice, very high incidence of the induction of ventricular tachycardia. And if those proteins are overexpressed, then without any stimuli, spontaneously, they're going to show the ventricular tachycardia. So expression of KLF15 is controlled under biological clock. This has been already known. And the expression of KLF15 is decreased in congestive heart failure. And KLF15 regulates the expression of the potassium channel, which causes the loss of QT interval, and it causes a ventricular tachycardia. So the clock gene abnormality may cause ventricular tachycardia. And I also show the another example. So this is about the cardioprotection. So there is a word says that ischemic preconditioning. So in the heart, if the heart is exposed to the repeated ischemia, the adenosine signal will be upregulated, which ended up in cardioprotection. So for example, this is IP, means ischemic preconditioning. So those are exposed to the ischemia. So repeated exposure to the ischemia, and after that, those mice exposed made the model of acute myocardial infarction. Then with the repeated exposure to the ischemia, infarctocytes or troponin I decrease. So this is cardioprotection. But part two, this part two means one of the important gene knocked out. Then those effects of preconditionings are diminished. From this, the authors discuss that this adenosine signal caused the stabilization of part 2, which led to the stabilization of hypoxic-inducing factor HIF1 alpha. And this caused the improved cardiac metabolism. And finally, this work for the cardioprotection. So the first question, will abnormal circadian rhythm worsen the prognosis of cardiovascular diseases? Although all are animal study, but there is a possibility that abnormal circadian rhythm may affect arrhythmia and cardiac ischemia from these studies. And let's move on to the second question. Will correction of abnormal circadian rhythm improve the prognosis of cardiovascular diseases? So this is very, very old study, there is a hamster which circadian rhythm is 22 hours, 2 or 3 hours shorter than normal. And those hamsters, the lifespan of those hamsters are shorter than normal hamsters. And to that special hamsters, when the normal SCN, the central clocks, are transplanted, then those hamster shows a normal rhythm. That means almost 24 hours rhythm are restored. And after those normal rhythm acquired, then those hamster can survive better than the abnormal hamsters. So that means normal rhythm is important for their lifespan. So we also tried to check the effects of the rhythm corrections. So we used cry knockout mice. Cry is one of the clock genes, and cry knockout mice lose the circadian rhythm. And we used those mice and wild type mice to measure the blood pressure by using the telemetry system. So we implanted radio telemetry monitoring system in the mice, and mice can freely move and record by radio telemetry the blood pressure. And in the wild type mice, as you can see here, the mice shows a very nice circadian rhythm in blood pressure. And here the black bar shows the dark condition, so without light. So all day long, even those mice are housed in this condition, but still they can show the nice circadian rhythm in blood pressure because they have a normal clock. However, cry knockout mice, which has no clock, they cannot show the diurnal variation or circadian rhythm of the blood pressure in constant darkness. So this was very predictable results, expected results. However, when those mice put in the light dark cycle, so from 9 o'clock to 9 o'clock in the evening in the light, under light, and from 9 o'clock in the night until the 9 o'clock in the morning, the mice put in the darkness. Since mice is a nocturnal animal, so their blood pressure increases in the night, subjective night. And in the light time, they tend to sleep. So they show the nice circadian rhythm. So in the case of cry knockout mice, when they put in the light dark cycle, then they start to show this kind of diurnal variations of the blood pressure, even though they do not have a clock inside. Why this happens? This is called so-called masked effect. So even without the clock system, when mice recognize the light and darkness, then those behavior or some of the physiological function start to make the circadian rhythm. So this is not caused by central clock. So when we check the heart tissues, we found that if the mice put in DD condition, which means constant darkness. DD conditions, the perivascular fibrosis or interstitial fibrosis were observed in these blue colors. However, if those clinoclot mice put LD condition, which has a rhythm, then those fibroses are much less than DD condition. So this data suggests the importance of diurnal variation of the blood pressure. So from other group study and our study, we would say that induction of circadian rhythm may suppress the cardiac dysfunction. And I would like to introduce other interesting paper, which is also very old, but they used our cardiac hypertrophy model. This is by aortic banding. So after aortic banding, they put mice in 24-hour light-dark condition, which means the normal pattern. So this black one is a monitor of the behavior moving. So these are night and these are daytime, which mice tends to sleep. So by aortic banding, the pressure overload, those perivascular fibrosis occur. And when these mice put in 20-hour light-dark condition, this is not different. This is totally different from this 24 hours cycle. And so you can see the behavior very strange. And when these mice are checked, then perivascular fibrosis is very severe as compared with this one. And when they put back these mice to 24 hours LD, normal pattern, then those behavior recovered to the normal. And this fibrosis get better. So aortic banding model, when they put in the abnormal rhythm, then abnormal behavior and abnormal expression of clock genes and cardiac hypertrophy and fibrosis are worsening. And when they put back in normal rhythm, then those are alleviated or normalization. So those are the rhythm adaptation appears to be very important. And this is the different study. Again, the abnormal hamster with abnormal rhythm. This is 22-hour cycle. When these mice, these hamsters are put in normal 24-hour cycle, so 12 hours light, 12 hours darkness, then they show these several abnormalities and short survival. If those hamsters are put in 22-hour cycle, which match to their internal rhythm, then it shows normal. So 24 hours is not important. The important things is that the synchronization of internal rhythm and outside environment. So asynchronization of internal rhythm and outside rhythm may cause the worsening of cardiac dysfunction or congestive heart failure associated with hypertension. So all of these are animal study. How about in human? So the shift worker, there are many shift workers, and some of you may work as a shift worker in night the night shifting. So all those shift workers cause be a irregular sleep and eating pattern. So they may have abnormal biological rhythm. So this is one epidemiological study from Japan, which is a study about the risk of shift worker. So about all-cause death, daytime worker or nighttime worker or shift worker. Shift worker means sometimes they work in daytime and sometimes nighttime, like nurses. So about all-cause deaths, it's not big differences. However, death by ischemic heart disease, shift work has much higher relative risk as compared with daytime worker and even nighttime worker. So what is the reason? Still, we don't know. But there is one consideration that, as I explained, the central clock control peripheral clock. And central clock is under the strong effect of the light. So the central clock and peripheral clock are synchronized. However, food intake may affect very strongly in the peripheral clock, especially liver and intestine. And in this case, the strong effect from central clock might be diminished by these effects. So that causes the shifting of the clock or dissociation of central clock and peripheral clock. So this is the very simple scheme that usually right and central clock and peripheral clock, but diet may disturb these hierarchy. So in the modern time, the convenience store or junk food in the all day long, and some kids enjoy playing game all day night, all night, and some school. All those abnormal rhythms may occur in the modern societies. So those may affect the future occurrence of the diseases. So this is the final slide. As a conclusion, the optimization of biological rhythm could be a therapeutic approach for cardiovascular disease associated with hypertension. And finally, currently in our laboratory, these members have been working very hard. And currently in the laboratory, we are not working on the clock study. Clock study was like almost 10 years ago. And most of them is now working the obesity or pulmonary hypertension and some other diseases. So next time, I hope I can introduce their work. Thank you very much for your attention.
Thank you, Emoto Sensei, for the excellent presentation emphasizing the circadian rhythm. Okay, we are going to move to the next session. To all the participants, we strongly encourage you to actively participate in this session by sending your question or your comment at any time during the presentation through the chat. Okay, I will hand over to the MC. Thank you.
Thank you very much for the interesting session, Professor. Now we are going to have our question session. We invite all the participants to fulfill the column chat to give some question that you want to ask for all the speakers. And if you want to ask directly to our speakers, you can raise your hand and we will collect all the questions in the last session. Thank you. Okay, as we continue to the second session. This session will be moderated by Dr. Dhite Bayu Nugroho, MSc, PhD. Dr. Dhite, the time is yours.
Thank you very much, Dr. Tabita, for the introduction. So now we are we would like to start the second session of the webinar, and we are very grateful that in the second session we will have Professor Tadahiro Kitamura to give talk on the role of glucagon. But before we start the second session, I would like to present the curriculum vitae of the next presenter. Yeah, thank you very much. Professor Tadahiro Kitamura is currently a professor in the Metabolic Signal Research Center in the Institute for Molecular and Cellular Regulation in Gunma University. Professor Tadahiro Kitamura earned his MD in the Internal Medicine from Kobe University and the PhD in the Endocrinology from Kobe University in 1996. From 1989 to 1990, he was a resident in medicine in Kobe University Hospital, Kobe, Japan. And then from 1990 to 1992, a resident in the Department of Internal Medicine, Hyogo Prefectural Kakogawa Hospital. From 92 to 1996, postgraduate student in the Department of Internal Medicine, Kobe University School of Medicine. From 96 to 99, he was a research fellow in the Department of Internal Medicine, Kobe University School of Medicine. And then he became a research fellow in the Diabetes Research Unit in the Division of Endocrinology, Department of Medicine, Columbia University from 1999 to 2005. An assistant professor in the Diabetes Research Unit in the Division of Endocrinology, Department of Medicine, Columbia University until 2006. And from 2006 until now, he is a professor in the Institute of Molecular and Cellular Regulation, Gunma University. From 2009 until now, he is a director of Metabolic Signals Research Center in Gunma University. And from 2013 until now, he is the director of Lifestyle Disease Analysis Center, Gunma University. And as we can see here, Professor Tadahiro Kitamura is a member of many associations that I may not be able to describe all. So before we start the second session, I would also like to remind all the participants that you can ask question. Doesn't have to be in English. You can ask the question in Indonesia also, and you can ask your question by writing it on the chat column. Or if you like to directly ask your question to the presenter, you can ask directly to the presenter at the end of the session. Okay, thank you very much. And I would like to invite Professor Tadahiro Kitamura to start presenting on the second session. The time is yours, Professor. Thank you very much.
Okay, thank you very much, Dr. Dhite, for kind introduction. And I also appreciate the organizers of this program in Gajah Mada University for giving me this opportunity to talk about my recent research. So let me share my slide. So just a moment. So can you see my slide? Okay.
Yes, Sensei.
Yeah, today I'm going to talk about glucagon, another important hormone for diabetes. So although Dr. Dhite already introduced me before starting my talk, let me introduce myself again. So actually, I was graduated from Kobe University Medical School in 1989. So this university is the same as the former speaker, Professor Emoto. So he is actually 2 years senior than me. So it's my pleasure to join this program together with Emoto Sensei. And now I am working in the Institute for Molecular and Cellular Regulation in Gunma University. So this institute located in Maebashi City in Gunma. Our institute was established in 1963 as an only one research center specialized to endocrinology in Japan. And this institute was selected twice as a Center of Excellence COE program supported by Japanese government. And at present, this institute is certified as a joint usage research program for endocrine and metabolism. And my laboratory is a metabolic signal research center in this institute. We are now focusing on elucidating the etiology and the pathophysiology of type 2 diabetes and obesity. Why we are focusing on these two topics? Because metabolic disorders caused by obesity, which we call metabolic syndrome, is known to increase the incidence of cardiovascular disease around twice. And also metabolic syndrome leads to type 2 diabetes. And type 2 diabetes has 4 times higher risk of cardiovascular disease. So as a result, the lifespan of diabetic patients with obesity is 10 years shorter than healthy people. That's why we are focusing on these two topics. And my present lab members are here. And Dr. Winda Aryani, she came from Institute of Technology Bandung in Indonesia around 10 years ago. So Fina, you may remember her?
Yes, I remember her.
You remember her?
Yes.
She is now working in my laboratory.
Oh yeah, very good.
Then let's go back to 2014. At that time, Dr. Fina Susanti was a PhD course graduate student in my laboratory. And
it's my pleasure to be in your laboratory, Professor. Yeah. Thank you very much.
Yeah. And she got a PhD in our institute. At that time, you were pregnant.
Yes.
And every year, we enjoyed year-end party around this season. It's a costume party. I hope you remember.
Yes, remember very well.
Yeah, actually.
Nice party.
Nice party. In 2012, I became Lady Gaga.
Did you also sing?
Yeah. And actually, 2 years ago, in 2019, just before the pandemic of COVID-19, I became a policeman.
Very nice.
Yeah. So, okay, let's move to a serious lecture from here. So, as you know, recently, incretin related drugs, such as DPP-4 inhibitors and GLP-1 receptor agonists, are globally used to treat diabetic patients. So these drugs decrease blood glucose levels by two mechanisms. One is enhancing insulin secretion, and another is suppressing glucagon secretion. On the other hand, metformin is the most popular drug in the world to treat diabetes. In 2013, published in Nature, the new mechanism how metformin decreased hepatic glucose production has been clarified. As shown here, metformin increase AMP level in the hepatocyte, and increased AMP inhibits glucagon signaling in the cells and decrease hepatic glucose production and decrease blood glucose level. So metformin decrease blood glucose level via inhibiting glucagon action in hepatocyte. By the way, compared to the insulin research, glucagon research has been delayed so far. The most important reason for delaying glucagon research is a difficult measurement of glucagon in plasma. By contrast to insulin, which is generated from proinsulin together with only C-peptide, glucagon is generated from proglucagon together with so many similar hormones called glucagon-related peptide, especially glycentine and oxyntomodulin are completely overlapping with glucagon. So regular glucagon immunoassays cross-react with these hormones, glucagon-related hormones. It's a cross-reactivity. So, and because C-terminal end of glucagon is unique and different from the other glucagon-related peptide, so conventional glucagon assays used this C-terminal antibody. However, it has been recently revealed that in human plasma, there are peptides having the common C-terminal end with glucagon. So even C-terminal antibody still cross-react with the other peptide. Therefore, to overcome this problem, recently a new glucagon measurement system, sandwich ELISA, has been developed. In this system, we use both C-terminal antibody and N-terminal antibody, like sandwich, and detect only glucagon specifically. However, when we conducted cross-reactivity test for this sandwich ELISA, we found this system still cross-react with the other glucagon-related peptide here by several %. I guess this is because either N-terminal antibody or C-terminal antibody is not completely specific for glucagon. So I think this is a limitation of immunoassays itself. Therefore, we changed the assay principle from immunoassay, which is based on antibody, to LC mass system, which is based on quantitative measurement with molecular weight of glucagon. So due to the limited time, let me skip the detailed method of this system, but we can say that this system does not have any cross-reactivity at all with the other glucagon-related peptide, because the molecular weight of the other peptide are totally different from glucagon, and this system measures glucagon by molecular weight. So totally no cross-reactivity. So we tried to apply this LC mass system for clinical examination of patient samples, but very unfortunately, due to two critical reasons, this system could not be used, could not be applied to clinical examination. One is that it takes 1 hour to measure each sample, so incredibly time consuming. And another reason is that the cost of measurement is much, much higher than immunoassays, like sandwich ELISA. So unfortunately, even we succeeded to generate very accurate measurement system of glucagon, we couldn't apply this system for clinical examination. Then we decided to use this LC mass system to evaluate the accuracy of various immunoassays, because immunoassays are less expensive and high throughput examination. So we compared the glucagon values between LC mass and the conventional competitive real or LC mass with new sandwich ELISA. And these are results. For example, as shown in the left panel, the glucagon values measured by conventional competitive real did not correlate with the glucagon values measured by LC mass, did not correlate well. By contrast, glucagon values measured by sandwich ELISA very well correlated with the result of LC mass. Compared to the left panel, the right panel shows much higher correlation. So we concluded that sandwich ELISA is a more accurate measurement system for plasma glucagon than conventional competitive real. Then, using this new sandwich ELISA, more accurate system, we investigated the pattern of plasma glucagon levels during meal tolerance test and glucose tolerance test in healthy people, not in diabetes patients. These are results in healthy people. So meal tolerance test are red line and glucose tolerance test are blue line. As shown here, blood glucose levels and the plasma insulin levels were almost comparable, almost similar between meal and glucose. However, the pattern of glucagon are totally different, like opposite between meal and glucose. The pattern of incretins, GLP-1 and GIP are also almost similar between meal and glucose. So interestingly, only glucagon showed different opposite pattern. So the question was why? Why glucagon showed opposite pattern? The most apparent difference between meal and glucose are that meal include protein and fat. So we speculated, either protein or fat stimulate glucagon secretion. So to verify this speculation, either protein or fat may stimulate glucagon secretion, to test this speculation, we conducted single nutrient loading test in healthy people. Single nutrient in the same healthy people. So one people has 3 different loading tests. One is glucose loading, and the second is a protein loading, and the third fat loading, and evaluated various parameters related to glucose metabolism. As shown here, blood glucose levels increased by only glucose, but not protein nor fat. And as to insulin, both glucose and protein stimulated insulin secretion. But fat did not affect insulin secretion. Very interestingly, 3 different patterns were observed in glucagon. Glucose decreased glucagon secretion, and fat did not affect it at all. However, protein strongly stimulates glucagon secretion. That's why meal increased glucagon secretion, even though glucose decreased glucagon secretion. Protein has a strong effect on glucagon secretion. So as to incretin GIP, all 3 nutrients stimulate GIP secretion. Then next question was, why? Why glucagon secretion was enhanced by meal intake, including protein? In other words, what's the physiological significance of postprandial glucagon secretion? Because usually in our meal, protein is included. So every time when we eat, glucagon secretion is enhanced. Of course, insulin secretion is also enhanced after meal, but glucagon also enhanced after meal. What's the significance? What's the physiological significance? So this slide shows the physiological function of glucagon in various organs. For example, glucagon suppress appetite and suppress gastrointestinal motility. And glucagon also enhance heat production, and glucagon enhance amino acid metabolism, especially ureogenesis. So it's quite reasonable that glucagon secretion is enhanced after meal intake, especially protein intake, to stop eating anymore and exclude ammonia produced by amino acid metabolism. However, one problem is because glucagon also enhances hepatic glucose production and increase blood glucose level. So from this point of view, it's very unreasonable that glucagon secretion is enhanced after meal intake. So still nobody knows the reason for this unreasonable action of glucagon after a meal. Then next important question was accurate glucagon measurement contribute to the diagnosis of diabetic stage and the decision of the treatment strategy. So to answer this important question, we conducted clinical study with type 2 diabetes patients. 19 healthy volunteers and 29 type 2 diabetes patients were entered in this study, and they were subjected to glucose tolerance test and meal tolerance test. Then evaluated various parameters related to glucose metabolism, including plasma glucagon levels. Glucagon levels were measured by sandwich ELISA and conventional competitive RIA. So first, I show you the results during glucose tolerance test. Red indicates type 2 diabetes patients, and the blue line indicates healthy people. Of course, blood glucose levels were higher in diabetic patients than healthy volunteers after glucose loading. And insulin levels were also higher in diabetic patients because due to compensative hypersecretion of insulin against insulin resistance in diabetic patients. So please pay attention to the left bottom panel. Glucagon measured by sandwich ELISA. This is accurate measurement system. At time 0, this point means simply fasted condition. Plasma glucagon levels was significantly higher in diabetic patients than healthy people. So fasted hyperglucagon secretion in diabetic patients. Additionally, at 30 minutes after glucose load, in healthy people, glucagon levels decreased. However, it was not changed in diabetic patients. Rather, a little bit increased in diabetic patients. There was a strong significance between these two groups. When we used the old measurement system, is a conventional measurement system, we couldn't see any change between these two groups. And as to incretins, GLP-1 and GIP, so not so big difference was observed between these two groups. So accurate glucagon measurement are more apparent to distinguish diabetic patients from healthy people. Then next slide shows data after meal tolerance test. Consistent with the data with glucose tolerance test, both blood glucose level and plasma insulin levels are higher in diabetic patients than healthy people, of course, after meal loading. And then glucagon measured by sandwich ELISA. Contrary to the data of glucose tolerance test, after meal intake, even in healthy people, glucagon levels increase. This is because meal include protein, and the protein stimulate glucagon secretion, even in healthy people. And the important thing is in type 2 diabetes patients, this increase after meal was apparently dramatically elevated compared to healthy people. There is a big significant difference. And by using the conventional old measurement system, we couldn't see such a difference between these two groups. And also GLP-1, GIP, not so, almost similar in between these two groups. So let me summarize these results here. We found three important significant differences in plasma glucagon levels between type 2 diabetes patients and healthy people. First, diabetic patients have higher fasted plasma glucagon levels than healthy people. And second, diabetic patients has no decline of plasma glucagon levels after glucose intake. The third, diabetic patients have the remarkable elevation of plasma glucagon levels after meal intake. So we concluded that accurate measurement of plasma glucagon levels is useful for the evaluation of diabetic state. And when we conducted correlation analysis, we found that the change of plasma glucagon levels from 0 to 30 minutes after meal intake, we call delta 30 glucagon. These values are very well correlated with various blood glucose related parameters. For example, fasting blood glucose level and blood glucose levels, the difference, the change of blood glucose levels from 0 to 30 minutes, postprandial blood postprandial blood glucose level, and area under the curve of blood glucose level, and also hemoglobin A1C, very well correlated. And also, we conducted histogram analysis. Histogram indicates the number of subjects as a y-axis, the number of patients or healthy people. And the blue bar indicate the healthy people, and the red bar indicate type 2 diabetes patients. And in the left top panel shows the data of delta 30 glucagon in meal tolerance test. So it's as a x-axis. So the more go to the right, the more plasma glucagon increased after meal. As shown, as you can see here, diabetic patients in red apparently shifted to the right. So many diabetic patients have higher glucagon secretion than healthy people after meal. So here, please pay attention to the blue line, a blue bar in healthy people. The highest delta 30 glucagon in healthy people was 35 here. And when we divided diabetic patients into two groups according to their delta 30 glucagon, higher or lower than 35, we can divide the group clearly into two groups. So we try to divide the diabetic patients. For example, red line indicates the people, patients whose delta 30 glucagon after meal was higher than 35. And the orange line indicates the patients whose delta 30 glucagon was lower than 35. And then we evaluated blood glucose level. And as shown here, in both glucose tolerance test and the meal tolerance test, blood glucose levels were significantly higher in the patients whose delta 30 was higher than 35, clearly indicating that the change of glucagon levels after meal well reflected their glucose tolerance levels in diabetic patients. Importantly, we also found interesting point that the glucagon abnormality in diabetic patients were not affected by incretin levels, because as shown here, when we divided the patients into two groups, their delta 30 glucagon higher or lower than 35, then evaluated GLP-1 and GIP, but we couldn't see any difference between these two groups. A little bit here, significant, but almost similar. So glucagon abnormality in diabetic patients are not related to incretin abnormality. More importantly, we also found that glucagon abnormality in diabetic patients are not affected by their insulin secretion levels. Because as shown here, even when we divided the patients into two groups according to their insulinogenic index, for example, insulinogenic index are lower than 0.4 or higher than 0.4. So insulin secretion impaired group and insulin secretions are of good groups, and evaluated glucagon values, but we couldn't see any difference between these two groups in diabetic patients. So these results are actually surprising to me, because many clinicians and researchers believe that glucagon hypersecretion in diabetic patients should be caused by impaired insulin secretion and following failure of insulin suppression of glucagon secretion, which we call paracrine mechanism. We believed so. However, our results clearly showed such paracrine mechanism does not exist in diabetic patients. Therefore, we can say that glucagon is a diagnostic marker for diabetes independent of insulin or incretin. We reported these results in Endocrine Journal last year. So if you are interested in glucagon, please take a look on this paper. Then the next question, important question, was does suppression of high plasma glucagon in diabetic patients improve the condition of diabetes? This is very important. And the answer is yes, because several glucagon receptor antagonists have been developed, and the effect of these drugs on blood glucose levels in type 2 diabetes patients have been reported. These are results. Glucagon receptor antagonist clearly decreased blood glucose levels in both fasted and fed conditions in a dose-dependent manner. Very beautiful dose dependency. So clearly indicating that when glucagon suppressed, blood glucose decreased in diabetic patients, regardless of their insulin levels. However, very unfortunately, this drug could not be applied for clinical use because, as shown here, many side effects were revealed in the phase 2 clinical study. For example, patients treated with glucagon receptor antagonist showed fatty liver and increased ALT levels and body weight gain and hypertension and hyperlipidemia. So many side effects. So the reason for these side effects are clear because many of the physiological functions of glucagon are beneficial for diabetes or obese patients. For example, glucagon suppress appetite, suppress gastrointestinal motility, and enhances heat production, energy expenditure, and enhance amino acid metabolism, such as ureagenesis. When glucagon receptor antagonist administered, these functions are blocked. And as a result, body weight gain, fatty liver, hyperlipidemia. So very clear. Even though, of course, this drug decreases blood glucose level. And so far, 9 types of glucagon receptor antagonists have been developed. And among them, 4 drugs are small molecule, and 4 drugs are monoclonal neutralizing antibody, and 1 is antisense oligo. All these drugs are effective for blood glucose level. However, due to the side effect, none of them has reached to the phase 3 study. So this is the last slide. So now in Japan, we can use 9 types of drugs other than insulin for the treatment of type 2 diabetes. Interestingly, the effects of these drugs on glucagon have wide variety. GLP-1 receptor agonist and biguanide metformin and the DPP-4 inhibitors suppress glucagon action. By contrast, sulfonylurea, SU, and glutamate and SGLT2 inhibitor and imeglutamine, these drugs conversely increase glucagon secretion. And alpha GI thiazolidine are neutral to the glucagon. So therefore, for the treatment of type 2 diabetes, we need to consider about the effect of these drugs on glucagon secretion. So thank you very much for your kind attention, and I'm happy to have your questions or comments. Thank you.
Thank you very much, Professor Tadahiro Kitamura. A really, really interesting talk that you just given regarding the role of glucagon in the diabetes mellitus sensei. So in the first session, you talk about the methods to diagnose or to measure the level of the glucagon and comparing the LCMS and also with the ELISA. We finally use the ELISA as a robust methods to quantify the glucagon, and then you also talk about the role of the glucagon to evaluate the diabetic state and also the role of several antidiabetic drugs to the glucagon, and the importance of also measuring glucagon and also considering the use of antidiabetic drug not only in the evaluation of the glucose level or in the insulin, but also the effect of these drugs to glucagon level also. So thank you very much, Sensei. Now we are entering the third session, which is the question and answer session. This question and answer session is a combination of the first session and the second session. So we would like to invite Professor Noriaki Emoto to join again the session to answer the questions. Again, I would like to invite all the participants to ask your question. You can ask the question in Bahasa or in English, and you can also write your question in the column chat or by asking directly to the presenter. So do we have questions already? Yeah, but before we read the question from the participant, we would like to invite the Dr. Vina. Dr. Vina is joining also the meeting, and Dr. Vina is the former student of Tadahiro Kitamura Sensei in his laboratory in Gunma, and Dr. Vina is the educational staff in the Department of Internal Medicine, and she is also the initiator of the working group Cardiometabolik. So you're holding a mask from maybe from
Yes.
Vina's from Vina-san, Sensei. Okay, so I would like to invite Vina-san to ask the questions. Please.
Thank you very much, Dr. Dhite. Yeah, thank you very much, Professor Emoto and Professor Tadahiro Kitamura for giving talk to this event. Yeah, it's very pleasure that we get the new knowledge of from Emoto Sensei and Tadahiro Kitamura Sensei. Yes, a very interesting topic that Emoto Sensei already explained about the circadian rhythm of the body. And you talk about the change of the circadian rhythm can influence the prognosis of the cardiovascular disease. And I'm also interested that I read about the circadian rhythm for the diabetes mellitus, maybe about the blood glucose level, cortisol, and also about leptin. Maybe do you ever read about this, and maybe what do you think about this circadian rhythm and with the blood glucose level and maybe leptin and maybe body weight? Is it also influence that maybe can be worsening the cardiovascular disease?
Thank you, Vina-san. Yes, of course, I believe that the circadian rhythm may affect many physiological and pathophysiological function, including metabolism or diabetes. And I myself am not working about the metabolic diseases, the diabetes, so I don't know exactly the current situation. But of course, cortisol is well known about circadian rhythm, and the disruption of those circadian rhythm may cause some disease. So, this may not be a right answer for your question, but I can just say that, of course, the circadian rhythm or biological clock may affect. This may not be the only reason, but this may affect in some aspects of the disease. Thank you.
Thank you very much, Professor Emoto.
Thank you. Dr. Vina, do you have any other question?
Maybe we can, there is also another questions from audience.
Yes, thank you very much. So now let's see what the audience have to say, or do they have any questions? So the committee, could you please show the questions from the audience? Yeah. Thank you. We have currently one question from the participant, Sensei. So this is the question for Emoto Sensei. Thank you, Professor Emoto, for the excellent presentation. I would like to ask about the circadian biomarkers chronobiomarker that can be used to monitor circadian rhythm? Also, what is your opinion about the cortisol in the pathophysiology of normal and abnormal circadian rhythm? This question is being asked by Novita Liman.
Thank you very much, Dr. Novita Liman. Yes, that's very important to find the biomarkers to reflect the circadian rhythm or to know the time for individual person. And we initially tried, and many people tried to check the clock gene expression in hematopoietic cells, like the neutrophil or some lymphocytes, and we could check the expression of those each clock genes in hematopoietic cells, but in fact this did not work. I mean, we cannot show a very clear circadian rhythm of these blood cells. And some people tried to use hair. I mean, by using hair, there is some cells, and they use those to check the circadian rhythm, but it didn't work. And some people use some cells from the mouth. So take out some the cell from the mice. This would not be so hard, and they check the circadian rhythm, and it appears to work, but still it's not established, the biomarkers. So getting the biomarkers would extend more knowledge about the circadian rhythm and human physiology or pathophysiology, but unfortunately, it's not available yet. Instead, melatonin is known to show a clear circadian rhythm, and since melatonin is strongly affected by central clock, so melatonin might be useful to see some circadian rhythm in human. Yes, and the second question about the cortisol, yes, as we discussed just before, the cortisol, the circadian rhythm of cortisol is very important physiology or pathophysiology. So, yeah, the loss of the rhythm of cortisol or may cause some or may worse to the disease status. Yeah, thank you very much.
Thank you very much, Emoto Sensei, for the clear answer. And let's move on to the next questions. I would like to invite all the participants. If you have any question, you can raise hand, and then we will allow you to directly, present your question to the presenters. Are there any participant who like to ask the question? Oh, yeah, there is one. Oh yeah, so this is a question from Dr. Bayu Hanggadi Nugroho. And this is question for Emoto Sensei, but before that, I would like to ask a question for Tadahiro Kitamura Sensei. Okay, Sensei, so in your talk, you mentioned about the role of the protein in stimulating the glucagon secretion, Sensei. So my question is, what is the actual mechanism, how the protein can stimulate the glucagon secretion? Is it by directly intrinsic mechanism, or is it indirectly? Although you also mentioned in your presentation that insulin is apparently not affect or does not affect the glucagon secretion, or protein can independently affect the glucagon, not by affecting the insulin, but there are several papers that mention about the indirect mechanism of meal or glucose in the glucagon expression levels, Sensei. So
Yeah.
could you please explain the mechanism of how protein or meal can stimulate glucagon secretion, Sensei? Thank you very much.
That's a really good question. And actually, we still don't have the answer to this question. The question is very simple, how protein stimulate glucagon, but still nobody knows the detailed mechanism, how protein stimulate glucagon. So of course, amino acid stimulate glucagon. And there are 20 kinds of amino acid, and many 17 out of 20 amino acid stimulate glucagon. But still, we don't know how, either directly or indirectly. Of course, everybody can guess, maybe indirectly. For example, paracrine mechanism, insulin or somatostatin may affect the glucagon signal, glucagon secretion. Or also, another important point is that neuronal regulation. Protein intake may stimulate some neurons and affect glucagon secretion neurologically, but still nobody knows the detailed mechanism. It's very interesting. It's very clear how glucose regulate insulin or glucagon secretion, but amino acid, protein, still not clear. So we need to do more study to reveal the mechanism. Yeah, very good question. Thank you very much.
Thank you very much, Sensei, for the clear explanation. And yeah, I think it's really interesting to see how the protein can, or the exact molecular mechanism of how protein can affect the glucagon secretion level, Sensei. Thank you very much. So
Dr. Dhite?
Yes.
Yeah.
Dr. Vina.
Can I say something?
Sure,
Yes.
sure.
Thank you very much, Professor Tadahiro Kitamura. I have same question with Dhite actually. So, but you already explained it. And based on that fact, how about the diet for diabetes mellitus patients? So, shall we limit the protein consumption for the diabetic patient, because the protein can, some of the amino acid can enhance the glucagon levels of in the patients.
Yeah, exactly. So another concept of this study is it's a diet therapy. So we need to think about the protein content in the meal for the treatment of diabetes, because protein content stimulates glucagon secretion and may increase blood glucose levels. So we need to consider about that. But recently, for example, many problems in the sarcopenia or frail, because all the patients have less muscle, skeletal muscle. So those older people need to take more protein. But old diabetic patients, when old diabetic patients take more protein, then protein stimulate glucagon and stimulate increase blood glucose level. So we need to think about more detailed nutrient content in the meal for the diet therapy of diabetic patients. This is a great point, very important point of Ina. Yeah.
Thank you very much, Professor Tadahiro Kitamura, because we also have many cases with the patient with hypoglycemia, recurrent hypoglycemia, and it is difficult for us to find the glucagon injection. So maybe we can also try to use some protein or amino acid to increase the glucagon levels, to increase the blood glucose levels, maybe, because here is the injection of glucagon is very difficult to get. Thank you very much. Professor.
Good point. Of course, in the condition of hypoglycemia, of course, glucagon is important to rescue the hypoglycemia in diabetic patients, of course. Yeah, this is also important point.
Yes, thank you very much, Professor.
Thank you.
Thank you very much, Dr. Vina and also Professor Tadahiro Kitamura. And now let's see if we have another question from the participant. Please, the committee, could you please share the slides, the questions? Yeah, thank you very much. So yeah, the next question is for Emoto Sensei. Thank you, Professor Emoto, for the great presentation. Yeah, I'm Bayu from Jakarta. I would like to ask you about the circadian rhythm and ventricular arrhythmia. I have two patients, female about 40 years old with frequent premature ventricular complex. The origin of PVC was identical from right ventricular outflow tract origin. But from Holter monitoring, the pattern of circadian rhythm of PVC is different in these two patients. One patient had PVC only in the daytime, but one patient had PVC appear in the day and the night time. How do you think about this phenomena? What caused the difference in the appearance of the PVC in these two patients? Yeah.
Thank you very much, Dr. Bayu from Jakarta. Yes, yeah, this is very interesting clinical cases. And I don't know the exactly, but I feel just this might be due to the difference of the relation between the arrhythmia and sympathetic nerve activities. So if it's only daytime, those PVC might be affected, strongly affected by sympathetic activity. And if it's not, I mean, if the PVC observed on both in daytime or nighttime, even in the sleeping time, those PVC might not be due to the, not related to the sympathetic or parasympathetic activities. And yeah, of course, the sympathetic or parasympathetic activity is strongly regulated under the circadian rhythm. So in other words, these might be affected by circadian rhythm, but there is no evidence yet that directly connect circadian rhythm and the occurrence of PVC, the daytime or nighttime. So yeah, I cannot give you a clear idea, but I just feel that this daytime PVC might be strongly affected by sympathetic nerve activity. Thank you.
Thank you very much, Sensei, for the clear explanation. And let's move on to the next questions. Is there any other question? Yeah. Yes. So this is question, but this question is being asked by Dr. Niken. And thank you, Professor Emoto, for the presentation. What is the most possible strategy to use chronotherapy in patients with disrupted circadian rhythm? What is the most possible and feasible strategy of chronotherapy to be implemented in patients with atypical diurnal and nocturnal variation? Thank you so much, Professor.
Thank you for your question, Dr. Niken Antea. Yes, again, actually, still, there is no clear answer, but from the experiments by using the rodents or animals, even though the circadian rhythm are disrupted because of the loss of clock genes or some abnormality of the central clock, when we put those mice in light and dark circumstances, then they would show some diurnal rhythm, although it's not complete, but some diurnal rhythm, especially in behavior and blood pressure and heart rate. This is not by clock, but this is so-called masking effects. So to restore or recover those rhythm, the sunlight might be a good inducer to get the circadian rhythm. And actually, for example, when you have a patient in the ICU after the deep anesthesia, they may really confused. And easiest way to make them the normal. In the morning, open up the curtain and expose them under the sunlight. Or there are some patients with dementia. They may walk around in the night, and they tend to sleep in the daytime. So in that case, expose those patients under the light. Then they may start to make some rhythm, and in this case, sleep-awake rhythm. Easiest way may be induce some rhythm by changing the light and dark cycle. Again, this is not clear answer, but thank you very much.
Thank you very much, Sensei, for the clear explanation. A simple question, Sensei. So, yeah. Is there actually any difference between the artificial light and the sunlight, Sensei, in terms of the regulation of the sleep and awake cycle, or light and dark cycle, or in the clock gene regulation, Sensei?
Yes, thank you very much for your question, Dhite-san. Yes, yes, there are some differences, and it's already studied a lot, but unfortunately, I did not follow very well, so I cannot give you the precise answer. But at least the light from the sun is very strong. And of course, the artificial lighting, of course, give you some effect for the clock gene, and in the rodent case. And again, in humans case, since we cannot monitor the clock in human, so we still don't know. But yeah, maybe you also experience that when you work in the night with the laptop computer or smartphone, the blue light may disturb your sleep. And this may affect the circadian rhythm or clock oscillation. But still, I myself don't know the exact answer. And it might be already elucidated. Thank you.
Thank you very much, Emoto Sensei, for very clear explanation. Again, I would like to invite a participant. Are there any questions from the participant for the presenters? You may ask directly to the presenters by clicking on the raise hand button. Questions? No? So yeah, before but while waiting for the question for the participant, I have another question for Kitamura Sensei and also for Emoto Sensei actually. So currently I'm working on the role of fasting actually, Sensei, the intermittent fasting, especially in the heart function. So we developed a heart failure model by using transverse aortic constriction in mice, and then we subject these mice to intermittent fasting, 24-hour fasting without food, and then 24-hour ad libitum diet. So my question is regarding the glucagon, and in my experience, in my research, we found a positive effect of these intermittent fasting in protecting the heart function under this heart failure model. So my question is for Kitamura Sensei, you explained about the protein role in the stimulation of the glucagon and also the glucose in the glucagon secretion. But how about fasting, Sensei? The fasting or fasting, what are the roles of this type of diet in the glucagon secretion, Sensei? Thank you.
So thank you very much for your question. Still, we don't know the effect of intermittent fasting on the glucagon levels. But of course, maybe circadian rhythm, of course, as Emoto Sensei said before, circadian rhythm are important, regulate glucose homeostasis in human, of course. So impaired circadian rhythm affect the glucose metabolism in human. But maybe the, regarding the hormones, circadian rhythm may affect more clearly on the cortisol or catecholamine compared to the glucagon, maybe, because the cortisol and the catecholamine has a circadian rhythm already. But nobody knows the glucagon, either whether glucagon has such a circadian rhythm in the physiological conditions. Still don't know. But so maybe we need to investigate this point, whether circadian rhythm also affect glucagon rhythm, but still don't know. Yeah, but I think good point, good question. Thank you.
Thank you very much, Kitamura Sensei, for the explanation. And maybe the same question for Emoto Sensei regarding the intermittent fasting. What is the role of the intermittent fasting, maybe in the circadian rhythm, Sensei? Do you have any? Maybe you read some articles regarding this, Sensei.
Thank you very much, Dhite-san. Yeah, I'm really interested in your study. Yeah, actually, first of all, intermittent food restriction is used in the study of circadian rhythm, but in this case, not 24 hours, rather daytime and nighttime, or like 8 hours fasting, or 8 hours food intake. As I explained, if the nocturnal animal provide food in their nighttime, this is daytime, so they have to get up and eat to survive. In this case, the central clock and peripheral clock are desynchronized. So the shifted the phase in the central and peripheral. But if it's 24 hours, I don't know what's going to happen for circadian rhythm. And if someone has already published or investigated, please defer those. And if not, maybe you can check yourself. And if it changed the circadian rhythm, and if those change in circadian rhythm affect the cardiac function, especially in the case of heart failure, that's really, really interesting. That may caused by the hormonal factors and the sympathetic or parasympathetic nerve system. Both sides may affect the heart failure. So, and in the case of the animal, you can dissociate those the pathways one by one. So yeah, I really hope that you can get the result and we can discuss it again. Thank you.
Thank you very much, Sensei. Now let's see if there are any other questions from the participant. We still have about 10 minutes, Sensei. And we have questions. Okay, could you please share the slides, the questions? Sorry, Sensei. Apparently, I'm still mute. So this is a follow-up question from Dr. Niken. Thank you, Professor Emoto, for the interesting exposure related to the reversible effect of fibrosis related to a proper regulation of light and dark milieu. So in such situation, is melatonin supplementation would be useful to be given for individuals with disrupted circadian rhythm. Circadian rhythm disruption also observed commonly in elderly. Should this type of population receive melatonin supplementation regularly? Please.
Thank you very much, Dr. Nikem. Yes, that's very interesting. And my answer is yes, but there is no evidence. There is no evidence, especially for human. But melatonin, not as a sleeping pill, which is now available. The pure melatonin called so-called Lamelteon from Takeda Pharmaceutical Company. So this is used for a sleeping pill to sleep, but those melatonin or this Lamelteon I believe that would change the circadian rhythm. So if one would take those melatonin or Lamelteon in the evening, then those people may get a good sleep and also kind of synchronize or induce the proper circadian rhythm. This is my guess or hope, but unfortunately, there is no clinical evidence yet. And so in the real world, we don't know. And the question, should this type of population receive melatonin supplementation regularly? I don't know if it's regularly or not, but at least if the sleeping patterns are disrupted, then for that time period, I think, it's not bad to give it a try to use melatonin. Yeah, thank you.
Thank you very much, Sensei, for the clear explanation. We still have a bit time for question. Are there any other question from the participant? Or would you like to ask directly? Please press the raise hand button. No? Okay. So yeah, maybe we are now at the end of the meeting, Sensei, at the end of the webinar. Again, we would like to say thank you very much, Sensei, for your participation in this webinar. And this webinar is the first international event organized by the working group Cardio-Metabolic in the Department of Internal Medicine. So we are very, very grateful and happy that you are that you agree to join this webinar, Sensei. And hopefully this webinar can be the beginning of a future and fruitful future collaboration between Department of Internal Medicine and Gunma University, and also Kobe University and Kobe Pharmaceutical University, Sensei. Okay, thank you very much, Sensei. Before we end this session, we have a photo session, Sensei. We have a photo session, so I would like to invite all the participants to turn on your webcam, and we'll have a picture together. Okay. Okay, on my mark, the first page, 1, 2, 3. Okay. Wait. Okay, 1, 2, 3. Again, and then on the second page, 1, 2, 3. Thank you. And then on the third page, 1, 2, 3. Ada berapa halaman? 2. Okay. Okay, Sensei, thank you very much. We can picture, and then yeah, I think with this I will end the webinar session, Sensei. Thank you very much, Kitamura Sensei, and also Emoto Sensei, and also Dr. Vina. Dr. Vina?
I'm sorry.
Yeah. Yes, Dr. Vina.
Yeah,
Please.
thank you very much, Dr. Dhite. Maybe I would like to say personally to Kitamura Sensei and Emoto Sensei. Thank you very much for the very interesting lectures, and we enjoy very much the fruitful discussion this time, and we get a lot of new knowledge about glucagon and also the circadian rhythm. And hopefully we can also do our next webinar in the better situation without pandemic, and we can meet offline in this webinar meeting. Thank you very much, Kitamura Sensei and Emoto Sensei. And I saw that Kitamura Sensei still keep my presence from Indonesia. Thank you very much for still keeping it. Yes. Thank you very much for everything, Kitamura Sensei and Emoto Sensei. I hope everything will be going better in these pandemic situations, and stay healthy and stay safe there. Thank you very much, Kitamura Sensei and Emoto Sensei.
Thank you very much.
Thank you very much. Thank you. Thank you.
Thank you.
Bye.
Bye. Dr. Dhite, can I ask a photo with Kitamura Sensei and
sure.
Emoto Sensei?
Yes.
For
sure.
Good memories.
Yeah, sure. Bisa dianting kita. Oh yeah, okay. Yeah. Let's take a picture on my mark. 1, 2, 3. Okay. Yes.
Thank you very much. So see you next time, Kitamura Sensei and Emoto Sensei.
Okay.
Please say hello to everyone in the laboratory.
Yeah. Thank you. Thank you.
Bye-bye.
Bye-bye.
Thank you very much.